Good morning, everyone. Thank you for joining us for today's webinar titled Expansion of Clinical Grade NK Cells from Diverse Sources to Therapeutic Potential. I'm Chingy from Atlantis Bioscience, and I'm delighted to be your host for this informative session today. Before we begin, allow me to do a brief introduction about our company, Atlantis Bioscience. Atlantis Bioscience is a Singapore-based life science distribution company established in 2016. We are a company dedicated to supporting researchers on their journey from bench to bed by providing high-quality translational solutions and services for several key R&D spaces. Beyond our role as a distributor, we also actively collaborate with professors and clinicians across the region, including countries like Indonesia and Thailand, helping to bridge the gap between academic research and clinical applications. These are our core areas of expertise: cellular and molecular study, where we offer premium cell culture reagents from BioS, Cogin Bio, imaging dyes from Biotium, and other reagents for omics studies. Cell and gene therapy is where we have one of the most extensive portfolios, ranging from the specialized media for stem cells and immune cells, AAV lentiviral vectors, mRNA LNP synthesis service, to exosome workflow solutions. For drug development and delivery, we offer a range of specialized cell culture devices and microfluidic plates, automated dissolution and diffusion system, and animal models. We are also active in the field of bioprocessing and alternative protein, where we offer batch reservation services for serum products dedicated for the scale-up production, and customized media specially formulated based on your culture needs. At Atlantis Bioscience, we carefully curate our product portfolio, selecting only trusted and proven brands. We are committed to ensure that the products that we distribute enhance your research efficiency, improve the experimental outcome, and ultimately accelerate the development of life-changing therapy. Without further delay, let us welcome our speaker today, Dr. Sergey. Dr. Sergey is the Immune Cell Therapy Advisor of our company. He's also the head of Immunotherapy Department at Bodros DOO. He was actively involved in COVID vaccine development in Russia, and he has over 10 years of experience in CAR-T and CAR-NK cell therapy research and development. Now I'll pass the control to Dr. Sergey. Dr. Sergey, you may share your screen once you're ready.
Morning, dear colleagues. Let me share my screen just a second. Okay, it's a great pleasure for me to discuss today the basics of NK cell biology and applications of NK cells, as well as expansion of clinical grade NK cells. So let's start. What is the core concept of NK cell activation process? So NK cells are tightly regulated by the balance of signals from activating receptors and inhibitory receptors on their surface. So if signals from activating receptors are stronger than signals from inhibitory receptors, then NK cell will activate and destroy the target. If signals from inhibitory receptors are stronger, then NK cell will just, go away and will not eliminate target, will not activate, will not proliferate. So when NK cell in our body meet normal cell, then a signal from MHC that ligate to inhibitory receptor on their surface is quite strong, and there is no signal from activating receptors on the surface, since normal cell do not express any specific ligands for activating receptors on NK cell surface. So there is no harm, no attack on normal cell by these NK cells. If NK cell is interacting with tumor cell, just typical plain tumor cell, and quite often we know that tumor cells are downregulating MHC on their surface. So NK cell do not have inhibitory signals from ligation of MHC with the inhibitory receptors on their surface, but still NK cell could have a strong signal from activating receptors, because quite often tumor cells are expressing ligands for activating receptors on NK cells, like MIK, for example. So after such interaction, NK cell will activate, will destroy tumor target, will start proliferation, everything is fine. Quite often, we have some kind of ambiguous situation when tumor cell express both ligands for positive signal and ligands for negative signal on NK cell surface. So NK cells could receive simultaneously strong positive signal and strong negative signal. What will determine fate of NK cell after that is the balance of these signals. If positive signals are a little bit stronger than negative signals, then NK cell will activate, will destroy target. If negative signals is just a little bit stronger than positive signal, then NK cell will not attack tumor cells. So this balance could be quite fragile. And in some cases, we could see that NK cell will not target tumor cells in patient's body. That's why in a number of cases, we have developing tumors in patients that is quite tolerable to the attack of NK cells. Every year, we have several cases of tumor cell formation in our body. And in most of these cases, immune cells and most NK cells will destroy these newly appearing tumor cells without our knowledge. So, of course, it's quite interesting, it's quite promisable to use this strong tool, these strong effector cells for therapeutical purposes. So what could be potential sources of NK cells for cell therapy? We could use autologous NK cells, so patient's own NK cells. We could use allogeneic NK cells from cord blood or allogeneic cells from peripheral blood of healthy adults. We could use iPS-derived NK cells or NK92 or other cell lines. So what are the pros and cons of each of the sources of NK cells? First of all, autologous NK cells, patient's own NK cells. Of course, it's the safest source of NK cells, because since we are using patient's own NK cells, we are not really worried about any potential infections. We are not introducing any other cells from other patients. That's also the cheapest source of NK cells in terms of QC, because we are not worrying about contamination with other cell types. If, let's say, we have contamination of T cells in our NK cell preparation, that's fine. We are not really worrying about graft versus host effect, because T cells of patient's own body is fine. You're just expanding patient's own T cells and then infusing it back. So no risk of potential side effects. The only problem of autologous NK cell therapy is a little bit questionable efficacy. We will discuss later the efficacy of different approaches of NK cell therapy, and we know that the majority of clinical trials weren't able to prove efficacy of autologous NK cell therapy for tumor elimination. Another source, we'll speak later about the reasons of low efficacy of autologous NK cells. But again, the reason is that strong signal from MHC could overweight positive signal on the surface of tumor cell. That's because patient's self-MHC will efficiently ligate with KIR receptors on the surface of NK cell. So in case of allogeneic NK cells from, let's say, cord blood, this source is also quite safe. Almost no risk of viral infections, because normally cord blood is safe from viral infection. The majority of virus can't penetrate from mother's blood to cord blood. And also extensive health screening that is employed for pregnant women in the majority of countries also help to assure that cord blood is free from any specific pathogens. T-cell contamination is not okay for this source, because we are infusing cells from other patient to other donor to our patient. That's why we have to be sure that we are able to eliminate the majority of T cells from our preparation. Also, it could be a bit hard to obtain cord blood in ethical GMP way, we have to be sure that we are able to establish a really good and ethical source of cord blood, that all patients are informed, are able to understand the purposes of cord blood donation, signed informed consent, and all other documents are in good shape. So the idea of the superiority of allogeneic NK cells over autologous NK cells is that we have so-called KIR mismatch. So inhibitor receptors on NK cell surface are not really good fit for MHC molecules on tumor cell, because tumor cell and NK cells are from different humans. And we know that NK cells express huge variety of KIRs on their surface, and each KIR could interact with some specific MHC molecules. So if we are infusing NK cell from one human to another human, when NK cell target tumor cell, the interaction between KIR and MHC could be suboptimal. With this suboptimal interaction, we are lowering negative signal that NK cell will receive from such interaction. By lowering negative signal from target cell, we are shifting balance toward activation and toward elimination of tumor cells. That's a simple way how we can improve efficacy of NK cell therapy with just this simple step. Just with switching from autologous NK cell to allogeneic NK cell, we are downregulating signaling from MHC ligands and therefore increasing activation, increasing efficacy of therapy. Allogeneic NK cells from healthy adults, also a good source of NK cells. These cells should be thoroughly screened for viral infection, because if we are using NK cells from healthy adults, we have to be sure that these patients are free from any viral infection. Potentially, we can have also some risk of side effects, because NK cell from adult blood could be a little bit more pre-activated, more mature, so potentially some side effects could be present, but still not really life-threatening side effects. iPS-derived NK cell is quite interesting and quite promising source of NK cell, because we know with iPS-induced pluripotent stem cells, we can obtain any tissues that we desire to use. So we can grow a huge bioreactor of iPS. We can then differentiate iPS into NK cells. So we can obtain big batches of NK cell, good quality, and NK cell could be manufactured from iPS derived from donor or from patient's own cells. So potentially iPS-derived NK cell could be a really good off-target drug, because theoretically you can manufacture billions and billions of NK cells with this approach, then cryopreserve these NK cells and use it like forever. So you can perform QC tests not for batch of, let's say, 1 billion NK cells, but you can perform QC tests for one batch of, let's say, 1,000 billions of NK cell. So you will decrease cost of your quality control dramatically, since you will be able to manufacture NK cells for hundreds of patients and perform QC tests only once for this huge batch. So that could be standardized, absolutely standardized NK cell therapy. The problem is that safety of iPS is still questionable. So we know that chromosomic instability of iPS potentially could shift this balance into the tumor-like cells. That's why a lot of companies are developing their iPS-derived NK cell therapy, and I'm pretty sure that sooner or later we will have some products on market that will be purely iPS-derived. But as for now, we should be quite worried about iPS-derived NK cell until we will be able to establish really good set of QC tests to be 100% sure that there is no chromosomic mutation in iPS cells that potentially will drive these cells toward tumorigenic field. Another interesting source of NK cell for therapy is NK92 or other cell lines. You know, NK92 is pretty popular NK cell line established from NK tumor. So that's basically cancer NK cells established from patient with NK cell nasal tumor. So these cells could proliferate at a really good speed. You can grow billions and billions of NK92 cells in bioreactor. These are suspension cells that are relatively easy to grow. So you can just put 100-liter bioreactor, grow a lot of NK92 cells, cryopreserve them and use like forever. It seems to be the cheapest source of NK cells for therapy. It's also pretty standard because it's a monoclonal line. It's quite safe. We know that several clinical trials were studied safety of NK92 cell therapy. Unfortunately, no efficacy shown today. Why we do not see any efficacy of NK92 cell therapy? I was also working a lot with NK92 cell line. We were also excited about possibilities of this cell line. We were making NK92 CAR cells. We were modifying the genome of NK92 cell. Unfortunately, NK92 cells have to be lethally irradiated before infusion because that's basically cancer cells. You know, you can't infuse living cancer cells into the patient. So you have to lethally irradiate NK92 cells before infusions to be sure that these cells will not proliferate in the patient's body. And unfortunately, when you irradiate NK92 cells, they will upregulate all the receptors typical for tumor cells. I mean, they will upregulate all the ligands, all the signals, please eat me, all the signals typical for foreign cells or irradiated cells or viral infected cells. Namely, they will highly upregulate MIC-A protein on their surface after this irradiation. So quite ironically, NK92 cells will be eaten by patients' own NK cells in a minute. When we were trying to establish mouse model, it's fine. You know, when you are using animals without immune system, NK92 therapy could be efficient. But when you're switching to patients, I know that one team of researchers in Canada were studying biodistribution of NK92 cells, and they were not able to detect NK92 cells after like 15 minutes after infusion. They weren't able to detect any NK92 cells in peripheral blood of the patient. So for now, I think the best option for cell therapy is allogenic NK cells from either cord blood or adult or healthy adult donor. So if we compare different sources of NK cells, we'll see that in terms of safety, definitely autologous cells are the best option. In terms of cost efficacy, NK92 cells are the cheapest solution because you can use just normal cheap media to grow billions of these cells. You just have to supplement it with IL-2, but it's also not too expensive. In terms of efficacy, allogenic NK cells from cord blood or peripheral blood of healthy adults are superior compared to other sources of NK cells for therapy. In terms of off-the-shelf availability, again, NK cells from allogenic sources is the better option because if you are using autologous cells, you have to take cells from the donor, then you have to manufacture NK cell product for, let's say, 2 weeks. Then you have to perform all the QC tests for sterility, for phenotype and potency, and potentially will take additional time. So you will be able to provide patient with GMP grade product only, let's say, in 3 or 4 weeks. If we are speaking about allogenic NK cells, you can just manufacture some batch of NK cells, without prior order. And then if you have patient, you just can infuse NK cells immediately after receiving order from this patient. So it's available as off-the-shelf therapy. Again, about iPS-derived NK cells, potentially that could be quite promising and interesting source, but as for now, we're not sure about safety and efficacy of this source. Okay, so for research purposes, I know that cord blood bags are available from Atlantis, from some certified European blood bank with donor GMP grade products. But to optimize your process, that could be a good start. Okay, what is the process of expansion of NK cells? When we are working with T cells, expansion is quite straightforward. We know that there is a major signal 1 and signal 2 for NK cell activation. Signal 1 is the signal from T cell receptor complex on T cell surface. Signal 2 is a signal from co-stimulatory receptor on signal surface. And signal 3 is a signal from cytokines. So when we are going to expand T cells, that's quite easy and straightforward. You're just mixing T cells with magnetic beads covered with anti-CD3, anti-CD28 antibodies, or it could be not magnetic beads, but dextrain or other specific ways to immobilize antibodies. So you're mixing T cells with these beads. You include cytokine in your media, and then you have really good expansion of T cells. You can grow billions of T cells from small amount of patient's blood, usually without any serious concerns. Of course, when we are working with blood from heavily pre-treated patients, when we are making CAR T cells, expansion of T cells could also be challenging, but in general case, expansion of T cells is easy and straightforward. Unlike T cells, and NK cells do not have some kind of common main zone switch. So you can't just put beads, you can't just trigger 1 or 2 receptors on T cell surface to induce their proliferation. So we have to play with the number of cytokines, and we have to employ special ways to stimulate NK cells. We were studying activating and inhibitor receptors on T and NK cell surfaces. And you can see here that we can see huge variety of activating and both inhibitory receptors on the NK cell surface, and not so much different activating receptors on CD8 and CD4 T cells. So for T cells, we have these key activating triggers, TCR and few co-stimulatory receptors. For NK cells, we have to trigger a lot of activating receptors on their surface to switch on these cells, to start proliferation, to activate NK cells. And in terms of biotechnology, that could be challenging. Historically, the first way for large-scale NK cell expansion was employment of feeder cells, so-called feeder cells. For this purpose, scientists modified K562 cells with the expression of special co-stimulatory receptors, ligands for example, 4-1BB receptor on NK cell surface, membrane-bound isoforms of IL-21, IL-15, in some cases IL-12, IL-18 cytokines, or some chimeric forms of these cytokines bound to the surface of feeder cells. Then you grow a lot of these feeder cells. You irradiate these feeder cells lethally to prevent their proliferation. Then you're mixing feeder cells with NK cells, and these feeder cells provide stimulation to NK cells, and you obtain good proliferation with a good speed and good yield of target cells. It could be easily 1,000-fold or 10,000-fold increase of NK cell after employment of feeder cells. Unfortunately, this approach is a little bit not convenient for biotechnology, because you have to be 100% sure that you don't have contamination of feeder cells in your end product. So you have to implement additional steps to your QC to be 100% sure that there is not even single viable feeder cell, because K562 cells are MHC negative cells, and you don't want to infuse even one MHC negative tumor cell in your patient that could be immunocompromised. Potentially, that could be really harmful for the patient. So you have to include a really sensitive assay to be sure that in your batch you don't have even single viable feeder cells. That's why when we are trying to decrease cost of production of NK cells, employment of feeder cells could be not the best option. Also, in terms of yield, in terms of proliferation, in terms of phenotype, feeder cells would be the best solution. So what could be the cheaper alternative to the feeder cells usage? We can immobilize special antibodies to the surface of the flasks. So these antibodies will cross-link activating receptors on the surface of NK cells. Then we will include cytokines to the media. Usually it's combination of IL-2 and IL-15, but optionally it could be also IL-21, IL-18, IL-12. You are using special media optimized for proliferation of NK cells, and you're using human AB serum. In the majority of clinical trials, researchers are still using human serum, at least several percents of human serum, because human serum is a great stimulator for NK cell proliferation. So after you switch on NK cell proliferation with this cross-linking of activating receptor, you can transfer NK cell to another flasks. I know that Atlantis is distributing specially formulated kit for NK cell expansion that include this agent for activation of flask surface with special agents, with special antibodies to cross-link NK cells, and include media for NK cell expansion. So you can just purchase this kit, and it's pre-optimized for the process of NK cell expansion. Most likely will still have to include human AB serum for the superior results. But again, you don't have to play around and to optimize density of antibodies on the surface of your flask and the manufacture of these antibodies. So if you're manufacturing NK cells from the allergenic source, then you have to pre-deplete your product to be sure that there is no T cells contamination. So usually protocol for NK cell expansion from allergenic source will be first you deplete T cells with the magnetic beads or with other approach. Then when you have PBMC already depleted, PBMC without T cells, you can proceed directly, let's say with the Atlantis Cogen kit, to expand NK cells with the T cell depleted PBMC as a starting material. Again, don't forget about QC for the NK cell end product. When we are speaking about autologous NK cell product, QC is a little bit more straightforward. You're just checking sterility, mycoplasma, potency. And when you're checking phenotype, more than 50% of CD56 positive, CD3 negative cells will be enough as the release criteria for your product. When you're manufacturing allergenic NK cell product, you have to be sure that there is no contamination with T cells. So the release criteria, you can fix that not more than 200,000 T cells per kilogram of patient's weight are present in your end product. So you have to be sure that T cell contamination is really low. When we are manufacturing NK cells, when we are developing protocol, we usually are able to obtain purity as high as 98% of NK cells, with T cell contamination as low as just, some fraction of percents. That's typical results from allergenic blood when you pre-deplete PBMC for T cells before expansion. So how about safety of NK cell therapy? Okay, let's take a look at the dynamics of clinical trials of NK cell products. we see that a huge interest for NK cell therapy grew just recently in recent few years, and more, not only phase 1 clinical trial, because we know phase 1 clinical trial, you're just touching this technology a bit, you're just trying to establish safety of this technology. But we see that more and more trials of phase 2 and even phase 3 are present in the current clinical trial landscape. So meta-analysis of safety of NK cell therapy shows us that there is no really risk of any serious side effects. So some potentially not life-threatening side effects like fatigue, nausea, anemia could be present for NK cell therapy, but it's only grade 1 and grade 2 adverse effects. It's not really too dangerous for patients' health. As for the grade 3 or higher adverse effects, again, only headache could be mentioned as a serious side effect, but that could be pretty typical for a number of for different kind of therapies, so it's not really serious and life-threatening. What could be the mechanisms of NK cell therapy? That's quite interesting. Let's take a look at the potential mechanism of NK cell therapy. First of all, let's check the efficacy of NK cell therapy. We see here that depending on the particular clinical trial, efficacy of NK cell therapy could be really different. You can see here a number of clinical trials with efficacy that is close to 0. You can see here some clinical trials with really high efficacy of NK cell therapy. So what is the key? Why the efficacy of NK cell therapy could be really could vary such dramatically from clinical trial to clinical trial? The reason is the source of NK cells and the method of expansion of NK cells. That's I put here citation from drug discovery paper, so you can see here that in general studies with unmodified autologous NK cell have yielded disappointing results. So autologous NK cell therapy usually will not provide with a really efficient remedy for tumor elimination. Again, another exemption from meta-analysis of different trials of NK cell therapy. And you can see here that for autologous NK cells, efficacy is usually not present. No tumor responses, no significant tumor responses, no tumor responses. Sorry, thyroiditis symptoms. So if we're speaking about NK cell therapy for wellness, longevity, beauty, whatever else, I know that a lot of companies in Southeast Asia are employing NK cell therapy for such application. Then we can't really speak about efficacy because there is no clinical trials to establish efficacy of NK cells for beauty and wellness, and I think it will be quite difficult to design such a clinical trial to measure efficacy of NK cells for beauty and wellness and longevity. If we're speaking about NK cells as a potential agent for tumor elimination, most likely we have to stick to allogeneic NK cells. We see here the results from one recent clinical trial of combination of allogeneic NK cells with pembrolizumab, and you can see here that if you compare pembrolizumab and infusion of NK cells, repetitive infusion of NK cells with pembrolizumab only, you see here significant efficacy of infusion of NK cells in combination of pembrolizumab. So in several clinical trials, we see here significant efficacy of NK cell therapy when we are using allogeneic NK cells as the source for as the source for NK cell expansion. So if we're speaking about therapy for tumor elimination, most likely we have to stick to allogeneic NK cells to be sure that there is some efficacy of our therapy. So what are the steps of NK cells in the process of tumor elimination? First of all, NK cells have to track to the tumor site, then they have to activate, then they have to lyse tumor cells, and then they can also activate bystander immune cells. So what could be potential challenges for all these 4 steps of NK cell tumor elimination process? First of all, poor NK cell biodistribution. We know that in some cases, if you have pre-established solid tumor, NK cells will not be just able to go inside to this pre-established tumor through the necrotic layers around these tumors. Then tumor cells could express ligands for inhibitory receptors on the surface of NK cells, and they could downregulate activity of NK cells. In some cases, tumor cells could be just lysis resistant, so you can't lyse them. In some cases, immune system exhaustion and tolerance would also play a negative role. Fortunately, we can partially solve these challenges. To better traffic NK cells to tumor sites, we can combine NK cell therapy with another types of therapy. For example, local irradiation. That could be a good idea to irradiate tumor locally, and then after tumor is already a bit inflamed, NK cells will better traffic to the tumor site. We can also combine NK cell therapy with the checkpoint inhibitors, so we can downregulate, downplay these signals from inhibitory ligands on the tumor cells. We can genetically modify NK cells. It's more challenging, but it definitely will be a good option. And we can also use checkpoint inhibitors to activate bystander immune cells. So I'm pretty sure that with combination of this approach, we can build better NK cell therapy, and we can have more and more happy and healthy patients. Thank you.
Thank you so much, Dr. Sergey, for the very insightful presentation. So I believe the audience would have some questions, so feel free to drop your questions in the Q&A box and we'll address your question accordingly. Let me check. Oh, I see we have a few questions here. So the first question would be, Dr. Sergey, what would be the challenges exist in maintaining the NK cell phenotype and activity during large-scale expansion?
Oh, thank you so much. That's a really interesting question. So when you are expanding NK cells and the big quantities, you have to first pre-optimize the process of expansion, because usually shifting in concentration of cytokines could dramatically change phenotype of NK cells. For example, we know that addition of some cytokines like IL-12, IL-18 could really shift NK cells towards so-called memory-like phenotype. And we know that more and more researchers believe that memory-like NK phenotype will be more beneficial for cell therapy. So first, I'm pretty sure that you have to pre-optimize your condition of NK cell expansion, and then most likely you have to include some additional cytokines, not only IL-2, but maybe, of course, IL-15 for NK cell proliferation, but maybe IL-18, in some cases IL-21, IL-12. So you have to play around with cytokines.
Okay, I hope Dr. Sergey has answered your questions. So we have another interesting questions over here. So in what ways do CAR NK cells offer advantages over CAR T cell therapies, particularly in terms of the safety, scalability, and also therapeutic potential?
I have some piece of unpublished data regarding CAR NK cell efficacy. We were making CD5 specific CAR T cells and CAR NK cells and compare efficacy of CAR T and CAR NK cells side by side. And we weren't able to detect superior efficacy of CAR NK cells compared to CAR T cells. So I would say that CAR NK cell topic, CAR NK cell field is a little bit overhyped, because in terms of safety, CAR NK cells is definitely safer than T cells. CAR NK cells also could be available off the shelf, and that's also another great advantage of CAR NK cell therapy. But if we are speaking about efficacy, most likely efficacy of CAR NK cell therapy will not be superior compared to efficacy of CAR T cell therapy. And when we check published clinical trial from Texas with the CD19 specific CAR NK cells. We see that efficacy is good, but it's not higher than efficacy of standard CAR T cell therapy.
Okay, thank you, Dr. Sergey, for the answer. Here's a pop-up message from Dr. Keep. I saw that you recommended the use of NK cell derived from cord blood from healthy donors. May I ask if there is any strong evidence of the efficacy available at this time?
Several clinical trials are now studying efficacy of NK cells derived from cord blood. By the terms NK cell derived from cord blood, I mean mature NK cells. I mean already differentiated NK cells derived from cord blood, because you can also differentiate NK cells from CD34 positive HCT, but here I mean just normal NK cells. We know that several clinical trials are exploring efficacy of such therapy just now. We don't have published data from clinical trials proving efficacy of NK cells from cord blood yet. But we see here that allogenic NK cells are usually more efficient in terms of tumor elimination compared with autologous NK cells.
Thank you, Dr. Sergey. So, another questions here. What are the hurdles in achieving the regulatory approval for allogenic NK therapies? So particularly, I believe some of you would like to know what is the current FDA expectation and what are the GMP requirements?
That's really awesome question. I like it. Well, that's also tough question. There is no FDA approved NK cell product yet to date. So we know that regulations for allogenic products are usually much tougher compared to autologous products, because you have to be sure that there is no any potential source of infections in your product. So you have to pre-screen your donor for a long list of infection, and then you have screen your donor again 1 month later. I also have to be sure that there is no contamination with T cells in your product. Usually, you also have to check MHC genotype of donor and recipient to ensure optimal or suboptimal fit. It depends on clinical trial setting, because in some clinical trials, we are targeting suboptimal interaction between carriers and MHC. We are targeting that MHC interact with carrier a little bit, but not too much. In some clinical trials, we know that researchers are trying to find as far as possible a source of NK cell as far as possible in terms of KIR mismatch to make it more active. So there is no any current regulation for allogenic NK cells, because we don't have any registered FDA registered NK cell product, but most likely it will be same as for other allogenic products. So you have, first of all, you have to be sure that there is no any risk of infections.
Yeah, very great answer, Dr. Sergey. Thank you for that. So we have another question in the chat box here from Anne. So is it possible that you can share some information about the transduction viral particles into NK to make CAR NK based on your experience? Like what vectors do you use? Any tips and recommendations?
Okay. Yeah. So in our experience, the best results is that when you're using a baboon virus envelope for pseudotyping of your lentiviral particles. So if, let's say, you're using VSVG pseudotype lentiviruses, you usually have to use MOI of, let's say, 30, 40, maybe 50 to ensure efficacy of transduction is high enough for your product. If you are using baboon envelope of the baboon virus, baboon leukemia virus, then you are able to use MOI of like 3 to 5, and still you will have enough efficacy for current case cell generation. The only challenge with this baboon virus envelope is that I know it's already patented. So if you're going to establish commercial product, you have to solve the issues with the potential usage of the IP by third parties.
So if any one of you is looking into, AAV lentivirals for any of your project, you may reach out to us because we are partnering with PackGene, providing custom service for lentiviral services, AAV, and also mRNA LNP services. There is one last question here, Dr. Sergey. What are the challenges in translating preclinical NK cell data to human trials?
Well, that's again really nice question, because we know that NK cells are relying on this, really accurate interaction between number of inhibitor receptors on their surface and ligands on the tumor cells. So when you are checking efficacy of NK cell therapy on animal models, you don't have ligands for the majority of inhibitory receptors in your animals, because in mouse you have another MHC molecules, you have another ligands for all these inhibitory ligands. And when you see high efficacy of NK cell therapy with the mouse models, usually this efficacy will not translate directly into the human models, because when you start infusing NK cells into the real humans, you will see that efficacy could be lower because of this interaction between inhibitor receptors on NK cell surface and MHC ligands in human body. So what we can do to target this issue is to perform more in vitro studies, employing tissues, employing maybe 3D spheroids derived from particular patients. Especially spheroid and organoid technology is quite helpful for in vitro discovery of NK cell efficacy.
Okay. Yeah, thank you, Dr. Sergey, for the answers again. I think that's all our Q&A session for today. So if you have any further questions, feel free to drop us an email. We will definitely pass your question to Dr. Sergey for any advice. So before we end this session today, here are some of the products that we offer for your NK research workflow. We offer human cord blood samples from CTI Biotech, Lymphoset, which is the lymphocyte separation medium to isolate PBMC, KBM NK kit that is specialized for NK cell culture mentioned by Dr. Sergey just now, GMP-grade cytokines to support your NK cell expansion and activation, and lastly, the flow antibodies for the analysis. So I'm proud to announce that we have also recently launched these two new products in our CGT portfolio. The first one would be HypoGuard. It is a serum-free and xeno-free hypothermic transport media that allows you to transport cells at hypothermic condition. It has been validated to be able to preserve cell viability of human MSC above 95% up to 120 hours. So here's the second products that we have. CryoEase protein-free version. It is a DMS salt-free and protein-free cryopreservation solution that has dual temperature compatibility in both hypothermic and sub-zero temperature. This allows you to maintain high cell viability and functionality of your sensitive cell lines by reducing the risk of ice crystal damage using a proprietary and stress-free formula. So if you are interested to explore more of this product on your sensitive cell lines like MSCs or even NK cells, kindly leave your email address in the chat and we will reach out to you shortly after the webinar. We understand that some of you might want to have a recap on our interesting sharing session today. We will send you a recording of this webinar and a series of related resources through email. If you have any further questions or you would like to discuss specifically on your research need, please don't hesitate to contact us via this email address. Other than that, you may visit our website to search for the resources that you need or browse our blogs for the latest updates of technology and tips. Lastly, don't forget to follow our LinkedIn page to get notified on our future exciting events and webinars. Thank you very much to Dr. Sergey again for your time and to our audience for attending our webinar. We hope that you have a very productive day ahead.
Thank you so much. Thank you
Bye.
for inviting me.